Researchers have made a major new advancement in creating synthetic life. This is so useful because, yes, it has a ton of biotech applications, but it also demonstrates that you don't need living components to make life, which is a little bit wild, and they went quite a bit further than previous studies. Yes, we know that we can take non-living components, mash them together, and turn them into a living cell. Like one of my favorites, of course, Mycoplasma laboratorium, the very first one, and research has been proceeding to make things like yeast, that is a eukaryote. It's more like us and it's many times more complex. This, however, involves taking the husk of another cell, so the one thing that we haven't really been able to do until now was to create a synthetic cell coat. This is called the cellular membrane, and it's composed of a lipid bilayer, but you don't have to remember that. You can take the DNA out of a cell and then put some new DNA in, and then all of a sudden you have a piece of life that has never before existed on Earth. You just have to assemble its genome. We have already demonstrated that the conditions on early Earth were such that you had all the ingredients for life, that you could make those lipid bilayers, and you could make things like ribonucleotides, but assembling them is a different matter. This research group did, named SPUD, and I can comment on the name all day, but that's not really what matters. They created that lipid bilayer in the form of a liposome, which is essentially a synthetic cell coat. It's used in our bodies to transfer all sorts of stuff, potentially DNA, and we aren't really sure exactly what they do, but they seem important. Either way, they can be turned into a living cell. It has 9 chromosomes, or at least plasmids. That's what chromosomes are called in bacteria. They're circular. We may actually have some circular chromosomes in our bodies, but they're not part of our main genetic code. Again, this stuff is very mysterious. We're still figuring it out. While this is just a proof of principle that yes, it can be done, you can create life, and I love that, they do have some work to do, because right now, SPUD is considered alive. It does have metabolism, it can reproduce for 5 generations, but it doesn't really have the complete genome necessary to make things like proteins. They need to give them the capacity to create ribosomes in order to make a functional organism. So technically alive, yes, it qualifies as alive, not very good at being alive, and that makes sense because this is something that did not evolve on Earth. It's something that we created. Given what we know about life, these are the genes that are necessary to create life, and turns out, yeah, you can. But why do we care? Yes, you can create life. That's neat. It tells us about our history on Earth, but what can you do with it? What we are demonstrating is that you can make programmable cells. You can make a cell that has not existed before on Earth that can potentially do a job. And this is important for a lot of fields, including that we kind of figured out we can make cells compute like a computer. Yeah, we can turn our cells into tiny computers. And that means that you could program cells to do things like chase down cancer. Really important. You could also reprogram the cells in your bodies to be younger, which is something that I've covered recently. I'm a little uncomfortable with the concept of turning cells into computers. I mean, what if they decide to do things that we don't want them to do and do it very efficiently? Although I am also kind of describing cancer. One of the things that I find interesting about this entire topic is how much people protest it, and it's kind of moving the goalpost, and I talk about the same thing with intelligence and AI, but it does apply to abiogenisis. Originally, people said that you could not make ribonucleotides, and then we did it. And then they said, well, you couldn't make ribonucleotides at the same time as a liposome, and then we did it. And then they said we could not assemble it, and then us humans did. And recently, I've heard people say that you need to stop borrowing the cellular membrane from another cell. And turns out, yes, we can do that too. So what's next? It seems like the limitation on this particular study is then giving it things like ribosomes and a cytoskeleton, so it can act more like a normal cell and stop being kind of a cruddy form of life. And that seems very possible too. Ultimately, where the goalpost will be is that, well, nobody saw life start for the first time, and that is true, and we probably will never see that happen unless somebody invents a time machine, and that is so far outside of my field. I'm a biologist. I am very excited for creating life, just like I'm excited for creating friends. Do I think all of it is a good idea? No, it's probably a terrible idea in some ways, but it is fascinating. I hope you've enjoyed this. I'll keep you up to date. Follow for more.